Hello, everyone, and thank you for joining us for today's webinar. I'm Heather Masaccio, your host from Bioprocess Online and Life Science Connect. With me today, I have Gina Kim, Director of Ab Technology Discovery Group from Samsung Biologics, here to introduce two new, highly effective ADC payloads for cancer therapeutics. After her presentation, we'll be taking submitted questions. Feel free to send these to us as we go along. We'll get to as many as possible during the Q&A, and any that we can't get to, we'll follow up with after the event. If you run into any issues, please try refreshing your browser first. And then if you need additional support, go ahead and submit a question via the Q&A, and I'll get us back on track. Finally, the webinar will be available for on-demand viewing. You'll receive a link via email as soon as it's available. With all that said, I'm going to turn it over to Gina to get us started.
Thank you, Heather. It's a pleasure to join this webinar. Hello, I'm Gina Kim, and I work in the Antibody Technology Discovery Group here at Samsung Biologics Bio R&D Center. Today, I'd like to introduce two novel linker payloads to support your ADC drug development journey. The agenda for the webinar is shown below. We'll start with a brief introduction to ADCs and their development trends and our solutions by offering two novel ADC payloads with a key result summary. Then we'll have a discussion at the end. Before discussing linker payloads, let's look into the ADC market. We see significant expansion in the requirement for ADCs, and moving from this year out into 2029, we're projecting over 2.5 times increase in the need for capacity for manufacturing ADCs. Of course, manufacturing these unique drugs requires a very in-depth understanding of how they work in their complexity. In line with the trend, Samsung Biologics is also exploring these technology trends and striving to secure innovation in the field. In this slide, we are going to look at the ADC development trend, and as you can see, the ADC type, which connects traditional anti-cancer drugs and antibodies, is the most developed so far. And the following remarkable item is multi-specific ADCs with a history of antibody treatments. The traditional ADC component mentioned in the previous slide is shown in the figure. The first component is antibody. Antibodies recognize target antigens and guide anti-cancer drugs to show efficacy only in specific areas, not the entire body. Second, the linker connects the antibody to the anti-cancer drug and prevents the anti-cancer drug from showing efficacy outside the target cell. Finally, the payload directly induces cancer cells to die, and it has various mechanisms. Payload plays the most crucial role in determining efficacy among the components of ADC. Look at the development state of various payload types. The tubulin inhibitor is still overwhelming as an ADC payload. However, the topoisomerase I inhibitor is the highest considering the number of active pipelines compared to the total number of pipelines. The next priority is the DNA damaging agent. As you can see, the results are similar even if only the pipelines that have entered the preclinical phase are collected. Still, the topoisomerase I inhibitor shows the highest ratio of an active pipeline. Tubulin inhibitors are most commonly used as payloads for ADCs, but limitations exist. Typically, tubulin inhibitors act on tubulin during cell division, so they are effective for rapidly dividing cancer cells, but have the disadvantage of being less effective for slowly dividing cells. On the other hand, in case of a DNA damaging agent, since it acts directly on DNA, it acts effectively on both rapidly and slowly dividing cells. From here, I'd like to discuss Samsung Biologics' approach and the next solutions in ADC drug development. We are internalizing ADC-related technologies to study and analyze its significant components, and in particular, this webinar will focus on two linker payloads, the camptothecin derivative and the duocarmycin derivative. Let me first talk about the camptothecin derivative among the two types of payloads being introduced today. Camptothecin is a topoisomerase 1 inhibitor that can be applied with high DAR and exhibits high efficacy in solid cancer. Camptothecin analog was initially unsuitable for use as an ADC payload due to its high lipophilicity, so we specifically designed the camptothecin derivative to make it suitable for ADC applicants. I will explain our camptothecin derivative by looking at more detail in result. When the bystander killing effect was measured by co-culture of HER2 positive cancer cells, SKOV3, and HER2 negative cancer cells, camptothecin derivative and exatecan derivative show similar results. In addition, we tested the immunogenic cell death to evaluate the possibility of combination with immune checkpoint inhibitors, which have been widely implemented in recent years. As a result, we confirmed that the immunogenic cell death rate of the camptothecin derivative is similar to that of exatecan derivative. In the right column, you can see the results of animal experiments. We conducted efficacy comparison tests using HER2 positive cancer cell lines and anti-HER2 resistance cancer cell line, and found that it showed equal efficacy. Next is the duocarmycin derivative. Duocarmycin is highly effective in all carcinomas, regardless of cell division rate. In addition, it is a weak substrate of MDR and PGP pumps. We stabilize this payload by site-specific conjugation and tumor-selective linker. We conducted a bystander killing effect assay, immunogenic cell death assay, and in vivo efficacy study. When the bystander killing effect was measured by co-culture of HER2 positive cancer cells, SKBR3, and HER2 negative cancer cells, duocarmycin derivative showed a higher bystander killing effect than DNA alkylator. In addition, our duocarmycin derivative's immunogenic cell death rate is also higher than DNA alkylator. In the right column, you can see the results of animal experiments. We conducted efficacy comparison tests using HER2 positive cancer cell lines and confirmed that the duocarmycin derivative showed superior efficacy than DNA alkylator. So we are continuing to develop ADC capabilities. Expected to be operational in December 2024, we have a dedicated ADC facility directly across from our main manufacturing site. We have single-use and stainless steel reactors up to 500-liter conjugation train and can deliver drug substances at a single site, providing integrated systems with established tools and procedures. All our facilities are clean rooms in Class C, and we are proud to have very safe ADC manufacturing sites. So, I want to wrap up this presentation by highlighting some of the ADC capabilities at Samsung Biologics. We offer comprehensive services that range from antibody production and conjugation development to manufacturing. We provide a high potential linker payload, the main topic of the webinar, which is easily accessible and cost efficient during ADC drug development stage. Also, we have a very dedicated ADC facility with dedicated labs and utilities for ADC and processes in cGMP suites feature isolators, and specialized equipment. Our target OEL level is 5 nanogram per cubic meter, one of the lowest in the industry, and we have systems to explore high potency compounds. ADCs have indeed demonstrated great clinical efficacy and occupy an increasingly important position in the therapeutic field. The success of the ADC is partly based on the stability and successful linkers for the delivery of the payload while maintaining efficacy and low toxicity. By our easy-to-access technology program, you can explore our 2 novel payloads for your ADC journey. So thank you for your joining our webinar today. This was Gina Kim from the Antibody Technology Discovery Group in Samsung Biologics. If you wish to explore our services more or discuss your developmental journey with us, please participate in our survey and tell us what areas interest you.
All right, Gina, thank you so much. What a fantastic presentation. I do have a few questions for you, so if you don't mind. First, this question is asking, can you tell me the stability of ADC equipped with Samsung Biologics linker payload?
In case of plasma, stability was confirmed, and the PK study will be completed by the end of November.
Okay, great. Thank you. Another question. Among ADC payloads, the efficacy of the drug is often low due to efflux. How about linker payloads of Samsung Biologics?
We have already tested it and confirmed that both linker payloads have low efflux. In the case of the camptothecin derivative, the experiment was conducted in comparison with exatecan derivative. And it was confirmed that it had low MDR and PGP pump sensitivity similar to exatecan derivative.
Fantastic. Moving on. I'm curious about Samsung Biologics' manufacturing capabilities. Can you provide anything other than CMO conjugation?
Samsung Biologics is building state-of-the-art ADC facilities to provide ADC development as well as manufacturing services. We can support lab-scale ADC PD and tox, material production in 20-liter scale conjugation scale.
And one final question as a follow-up to that, what is the possible range of ADC process development?
Of course, the development of ADCs has been complicated by the need to balance drug efficacy and safety and requires extensive investigations and experiences. Through our diverse experiences with conjugation technologies such as chemical and enzymatic conjugation for random and site-specific ADC, we have the capabilities to reduce unguided DAR specifications and increase homogeneity.
All right. Thank you so much for answering those questions. There's a lot of good information there. Thank you, Gina, for your time and to our audience for joining us today. If you'd like to contact Gina, her email is available in her speaker bio at the top right-hand side of your screen. We hope to see you again soon. We hope you have a great rest of your day. Take care.
Thank you.